So no matter what kind of cell you're thinking of, either a prokaryotic cell or eukaryotic cell, a lot of DNA is packed into a cell. And in bacteria, you typically have a single double-stranded DNA chromosome that's circular, and then some additional plasmid DNAs. And the chromosome will be attached to some proteins, sometimes called scaffolding proteins, and looped back and forth and held by those scaffolding proteins in a part of the cell called the nucleoid region. In this picture, they show a cell that has been broken open to release its chromosome. And but it's very tightly coiled. Sometimes that's called supercoiling. And so the very tight coiling of the DNA is important to pack a lot of it into a small cell. In eukaryotic chromosomes, the DNA is arranged in a little more regular arrangement with proteins. The 2 nanometer wide DNA double helix is wound around proteins called histones, and the histones are in groups of 8. 8 histone proteins make up what's called the octamer. The DNA wraps twice around each octamer, as you can see here, and it forms what's sometimes called the beads on a string structure. And there's 4 types of histones that are in the octamer, and then there's another type shown here like in yellow. But the beads on a string is then coiled around itself in what's called the solenoid structure, and that makes what's known as the 30 nanometer fiber. And then the 30 nanometer fiber is wrapped back around itself to make the 300 nanometer fiber. And then the 300 nanometer fiber is coiled around itself again to make the 250 nanometer and ultimately 700 nanometer structure, which is the chromatid. And this shows a replicated chromosome that has a 1400 nanometer diameter. So that'd be a human chromosome, but other eukaryotic chromosomes will have the same fundamental wrapping process. And so when DNA is fully condensed, it means it's reached this endpoint of the replicated chromosome. When DNA is decondensed, it doesn't unwind all the way. It only unwinds to about the 300 nanometer level or so. So DNA doesn't fully decondense all the way, for example, back down to just the double helix. But even in interphase, the chromatin will still be wound partially. But by prophase and metaphase, it fully winds to the structure shown here at the bottom. One thing that they have done in order to study chromatin structure is to use enzymes to break apart the nucleosomes. The nucleosome would be the histone octamer with 2 loops of DNA around it. So by mixing DNA with nuclease enzymes, the nuclease enzymes will cut any exposed sections of DNA called linker DNA, and then they can measure and study not only the nucleosomes but also just the histone octamer. So you can see here in this space-filling model, the histone octamer is composed of 2 molecules of histone called histone 2A, 2 molecules that are called histone 2B, and then it's not shown here, but there's 2 molecules of histone 3 and 2 molecules of histone 4. And then histone 1 was actually shown on the previous slide as that yellow histone that's sort of stuck to the linker DNA, so it's not part of the octamer. So I'm going to there's a link here that is a very helpful video on YouTube that shows the coil structure of fully condensed chromosomes. And I will put a link after this lecture segment. There will be a link that you can click on to watch that YouTube video.